all of you a warm welcome to this November edition of Sugar Rush with Science. Most of you here are already familiar with the event, but in interest of any freshers or newcomers who are here, Sugar Rush is a platform that is meant to give the campus community an opportunity to come together informally and share their recent research pursuits. And the aim is basically to increase campus dialogues, and that is across labs, across domains, across departments. Give an opportunity to understand the very diverse science and innovation that is happening across the cluster, and promote collaborations so that we are together as a cluster, we take more science to society. So a few housekeeping rules. So the format, if you are already familiar with it, it features two speakers, one of them being an academic researcher from the cluster and one an innovator or startup associated with CCAM. Both speakers, actually, because we want to keep it informal, we present it in chalk talk format. Both speakers have 15 to 20 minutes, then followed by a 5-minute Q&A. And to avoid a distraction to the speakers, we will unfortunately not serve ice cream while the talks are going on. You will have to wait until the talks are over to get your share of ice cream. So with that, I am delighted to introduce you to our first speaker of the day, Hitesh Goswami. Hitesh is the CEO and co-founder of 4baseCare. 4baseCare is a pioneering company in the field of precision oncology. Their vision is to make personalized cancer care more accessible and affordable, and with a focus on genomics-based equity in cancer care. And they are at the forefront of data-driven genomics-based biomarker testing in cancer patients in India, and they have recently launched state-of-the-art labs in Dubai, Philippines, and India and Nepal. And 4baseCare was the first company in the Asia Pacific to be selected for the Illumina Accelerator cohort in 2019, and they have recently raised Series A funding of up to $6 million from deep tech VC Yali Capital. And so Hitesh is here, and his talk is says scaling deep tech, the lessons on the road to Series A, and hopefully he will share not only his lessons on the road to Series A, but also a little bit about what is genomic data equity. Thank you.
Thank you so much. And thank you CCAM for providing this opportunity. So of course, thanks. I don't have to introduce myself and the company and directly get on to the talk. So we live in the era of data and personalization. Do we all agree? Right? More or less, data and personalization. And it's good in a way and bad in a way. It's good, like if Swiggy knows I like biryani, it gives me offers, it shows me any new kind of biryani has come. But it's bad in a way where if I'm talking to my daughter, teaching her about fish, and then suddenly I get messages from Delicious and Fresh to Home, right? So it all depends on how we look at the data and how this is being done. And especially in healthcare, I think we are more and more moving towards personalization. And with the advancement in technology and accessibility to technology like genomics or even data science, AI, machine learning and all, I think healthcare in the next decade is going to be in a different space altogether. The way conventionally treatment used to happen, everything is moving to a more personalization or precision, right? Even there's a lot of debate. There are people who think that AI is going to replace humans, or some people think AI is absolutely stupid, right? But I think AI or machine learning, it's essentially going to make smart people smarter and dumb people dumb. Right? So if you're smart, if you know how to do AI will actually help you in becoming much more smarter. So with that, we understanding that data is the important part of all these things. As a company, 4baseCare, what we realized is that, especially in cancer, which is the disease of the DNA, unfortunately for us to work on any kind of personalization or develop any kind of personalized solutions, there's not enough data available. If you look at the Cancer Genome Atlas, one of the largest database for cancer data, More than 90% of the data is from the Caucasian population. Overall, also some of the largest projects which are going on globally. One of the largest funded project called Genie, again in US, again working on the cancer data. If you look at it, the data diversity is one of the biggest challenge that particular project is facing. And at 4baseCare, we also realized this is a problem, and this is the problem that we wanted to solve. And again, data why? And why cancer essentially? Because cancer being the disease of the DNA, if you have more data on the DNA, understand the disease better, it helps us personalize the treatment better. And what we realized is that the way cancer treatment is, landscape is changing in the West, and where we are, there's a huge gap. What was happening even 10 years back is most of the patients, they would have a very personalized approach. So when I say personalized approach in cancer, what I mean is, for an oncologist earlier, he or she would just need to know where the cancer is, what stage it is. So if it is lung cancer stage 2, 100 patients coming, all of them would get a started regime of treatment. Now what has happened because of advancement again in genomics and data science, today we can stratify cancer patients into different subgroups based on the kind of mutations that we identify in the tumor DNA. So today, if I have an EGFR positive patient who has mutation in the gene EGFR, I give a treatment with the kind of drug called tyrosine kinase inhibitors. If there's a patient where, say, a PDL1 expression is high, we would give immunotherapy, right? So that way, every patient, the treatment changes. If you give immunotherapy patient a TKI, the patient is not going to respond, or a TKI to immunotherapy patient, the patient is not going to respond. And this is the way now the treatment paradigm is changing. And unfortunately, whatever solutions which were available in India were mostly developed based on the Caucasian data. So most of the diagnostic companies in India, they would buy these cancer panels or cancer kits, and they would provide the service and give the report back to the doctor. He said we need to change that, because firstly, overall, we are different. And we know that. I don't think I need to give importance on why we need to do this, but we need to do this, we need the data. Even if you look at EGFR mutation itself, very interesting numbers, statistics. So if in EGFR mutation frequency in lung cancer patient in Caucasian population is 15%, whereas in India it is 35 to 45%. So almost 40% average of your lung cancer patient will have EGFR mutation, right? So since there we can see so many differences, we decided that we are not going to use the available data, we are going to generate the whole data, and that's where Illumina Accelerator happened for us. We became the first company from Asia Pacific to be selected by Illumina for the Accelerator program. This was way back in 2019, and they funded our complete project. We said we are not going to touch your TSO 500 panel. We want to develop something on our own. So they funded our whole project, where we generated whole exome DNA and RNA from FFP. FFP itself is very difficult even with DNA, but we decided that we'll take out the challenge, even RNA will take it out from FFP and work towards generating whole exome data. And that's where we generated data from 1500 plus different cancers. So today we have close to 3000 FFP whole exome DNA RNA data with us. And from that data, we identified around 1200 genes which are associated with cancer, prognosis, pathogenicity, treatment, other things. And we developed world's first cancer gene panel, what we call world's first Indian population specific cancer gene panel, that we call Indiaga Gene Panel. And we launched that in 2021. So today we are the only company which has developed a population specific cancer gene panel. I'll tell you what impact is that now. So tumor mutation burden is one of the biomarker for immunotherapy. Tumor mutation burden simply means if I'm looking at the mutations in my data, how many mutations per MB of my region of DNA, right? Mutation per MB. That's how it's calculated. And TMB 10 plus. Greater than 10, essentially, if any solid cancer, be it breast cancer, colon cancer, lung cancer, pancreatic, prostate, any cancer, solid cancer, if the TMB score is more than 10, then FDA approval is there, where immunotherapy can be given to the patient. But what we have seen is less than 20% or even less than 15% of the patient respond to immunotherapy based on this particular biomarker. Now, is this the fault of this biomarker? No. What we realized is that firstly, across the cancers, you cannot have 10 as a universal number. The cutoff would be different in pancreatic cancer, the cutoff would be different in breast cancer, the cutoff will be different in lung cancer, so you cannot have 10. In fact, head and neck cancers, we see hardly any patient about 10. Mostly it's less than 10, right? And moreover that, on top of it, it's not just across the cancer types, but across the population. Also this would change. A 10 TMB score in an Indian lung cancer patient might mean something different compared to a 10 TMB score in a Caucasian lung cancer patient. So we did this research. In fact, we published in JCO, which is one of the ASCO journals, where we have first time we have shown the tumor mutation burden score in association with population and the cancer type. And we came to one hypothesis where we said, rather than giving just a score, so typically how it would happen is if it's a report, cancer report which has been given to a doctor, they will just say TMB and put the value. So say TMB 15.2. Now doctor has to decide whether I should give the treatment, immunotherapy, should not give the immunotherapy, because no diagnostic company will take the responsibility. What we thought is, what if we can give additional information? Of course, for us to prove a hypothesis, we need to do a lot of work more. But we did have a hypothesis where we decided that let us give doctors more information. Let us plot this. So say if I have pancreatic cancer patients, 500 patients, I have done the TMB analysis. Let me plot this data and make a percentile distribution. Okay, and then see where does 15.2 fall. So say this is pancreatic patient cohort with tumor mutation burden score, say 20, 40, 60, 100. So rather than just giving this number, what we started doing now is that 10 in pancreatic cancer patients in our cohort, Indian cohort falls where, so say it is falling here, 10, which is a universal cutoff for immunotherapy. And then we'll say where will 15.2 fall. So if it falls over here, right, means it's more than close to 90%. That means only 10% of the patients have TMB score more than 15.2. But these are just the random numbers I put. They are not the actual number, just for you guys to understand. Now this, as a doctor now, gives me understanding that if I just given say 12, right, and 12 is more than 10, I should just straightforward go for immunotherapy. But if it is falling somewhere here at 60 percentile, that means 40% of the patients are going to have TMB score more than 12. Does it make sense? Is this really too high TMB? We don't think so. And based on some of the data, retrospective data, where we have this information and we have the outcome information also, what we saw is that patients generally above 85 percentile are responding better to the treatment, right? And that's where we got a very interesting case, and this is where I'll show you the impact of having our own data and how it can impact patients' life. So we had a patient, head and neck cancer. 1 year, the patient was not responding to treatment. Maybe if there's a table, I can just put my laptop. Because this I want to show you guys. It's the actual case. Otherwise, I just told it. No problem. I just told it like this. Yeah. You guys can see? Of course. Let me just put it like this. Okay. You guys can see, right? This is a case that we had. Thank you so much. The parotid cancer, cancer of the salivary gland, falls into head and neck category. 1 year, patient was on standard chemotherapy. The tumor was progressing, no response. The doctor decided, let us see if we can get some more information on that. They sent the tissue to us in our lab. We did the whole workflow, RNA and RNA extraction, sequencing, bioinformatics analysis, and you can keep it down. I'll call you again. Thank you. Because now I need to use the board also. Now we did all these things, and we got a TMB score over here of around 5.8. Low, right? And then we went back to our data. We said, let's plot this in head and neck. it was coming at 95% time. Okay, so now we have not done any clinical study. We have no data right now. We just have hypothesis based on whatever analysis interpretation that we have done and the data that we have collected. And we had retrospectively, we had seen some of the patients where we had the treatment outcome data also, and what kind of TMB scores are there in these patients. And here the doctor was out of option, the patient was out of option. So we spoke to the doctor, we said that this is our internal data. So doctor is like, any which way this patient is not responding. If I don't do anything, the patient is going to die in maybe 6 months, 1 year, or 2 years. Let me try this. I'll talk to the patient and their family and see what is happening. So the doctor decided to go for immunotherapy based on this data. And this is the response. This is the response in just 3 months' time. There's no surgery. And this was 1 year later. And even just 3 months back, we had spoken to the patient again, still in complete remission. This is the impact. This is the impact. This is what data can do. This is what personalization can do. This is how we are going to manage disease as complex as cancer. But it's very important in terms of how ethically this data is used, especially when it comes to healthcare. And that's where I think a lot of discussions need to happen within the groups, ecosystem. Everything has to be matured enough to see how this data can be used, and a lot of collaboration is required. So 4baseCare as a company, it has two meanings. One is the 4 bases of DNA, but 4 base also means the 4 bases of cancer care. That's how we put it. ATGC again. So C, of course, is the clinical care that patients get in hospital with the doctors, right? T is the technology, like genomics and all. So the technology companies, technology partners who play an important role. G is the global or genomics research that supports what we do at NCBS. So right from coming there to getting into translational globally, that kind of research is required, right? And A is the light care that is provided, which is non-clinical. Patients need psychological counseling, nutritional counseling, they need financial support, they need that ecosystem where they can find all those things. So as a company, we wanted to bring all these together because they're all in silos. So another thing was that is how the ATGC 4baseCare came. We wanted these bases to come together. And that's how the company started. Then we started the company, I think, already a product introduction to the company. We started in, now just talk about how we as a company has grown from startup. For those who are startup over here, I think I've done the science part, if that is enough. So we started in 2018, started operations early 2019, and then immediately in 6 months, Illumina happened for us. So me and my co-founder, we both had met at our first work. We were working as research scientists in a drug discovery company in Mumbai. He was part of the team which was working on cancer vaccine development. I was part of the team which was working on screening and developing new assays for screening for anti-cancer, anti-diabetic, anti-inflammatory drugs. And then he went ahead and did his MBA from XLRI. He was part of G Healthcare team before we started 4baseCare. I got excited about genomics and I got excited about business overall. So I moved from doing lab work, got into application side and selling science, I would say in a very crude way, right? But of course my education in science and my lab work and everything of course helped me because I could understand to whom I was selling because they are scientists at the end of the day, or doctors in healthcare. So we both decided to start 4baseCare 2019, just 4 people company. Then we got our first pre-Series A funding, that was in 2021, we raised $2 million. And that time we were again around 15 people company, and now we are close to 200 people company. We just closed our Series A round of 6 million. We are now expanded to operations, so now we have a lab in Dubai. So we are looking at Middle East as one of the market. We are looking at Southeast Asia, so we have a lab. We are going to set up a lab in Philippines, mostly in January. Next month we are setting up a lab in Nepal. So what we realized is that the technology has to be taken to the patients. We cannot be a lab in India, Bangalore, saying that all the samples will be processed here. You have to go close to where your patients are, right? And work with the ecosystem over there, because every country would have their own data problems. They will have their own data solutions. So we need to work with the healthcare ecosystem over there. But having said that, it sounds all fancy. 1.5 years back, we had a call with all our team members saying that guys are not able to raise funds. It's a very difficult situation, and we need to take pay cuts and all those things. And at some point, we're also thinking of how do we exit. Maybe we get acquired, because we already have a panel now, work with some big diagnostic company, get ourselves acquired and all. So startup, it's not all fancy that looks outside. There are a lot of struggles which goes on, but I think you have to get going. Just go on, go forward. Things work out many times, most of the times. So there will be difficult situations in startup. You need to keep on going, and especially if you're in deep tech or research kind of a startup, things are going to be more difficult, because it's very difficult to explain to investors what you are doing and why you are doing. And the cycle of due diligence and everything is very, very long. So yeah, that's, I think, I don't know how much time is there, but yeah, pretty much I'm done with the talk. I'll be happy to take any question. Maybe I would like it to be more engaging. If there are more questions, that would be there. Thank you so much. Thanks everyone.
Questions? Sure.
So you were talking about these panels, right? So when you constructed this panel for your Indian cohort, how different did it look from the cohort, the panels you?
In fact, also. Hello. So we in fact also found some novel fusions. So we have patented 39 novel fusions that we found from this data, and now we are further validating them to see the application. So there's a lot of difference in the type. See, many of the places, I give you example, BUS is one of the category in annotation of these variants. You have clear family history. You have all the signs of this being a hereditary cancer in some way or the other, but annotation is coming at variants of unknown significance. So this data now, what we are looking at is also from this perspective, where it's not just the genomic data. See, genomic data on its own does not have value. Now today, our data is starting to gain some value because we have outcomes data. And that outcome data is something that helped us over here. So what we are looking at is not just the genomic data, but also looking at the clinical outcomes data to get more better interpretation than just what is the mutation, where it is, which gene it is, which coordinate it is. We are more looking from that perspective.
One more question I had is, are you also working on the resistance side? So what happens when, let's say, you're giving some TKIs, first generation, and then you get drug resistance. So do you have panels for that as well?
Panel will remain the same.
Ah, the panel.
But your the mutations which are associated with resistance, or the genes which are associated with resistance, are different than the genes which are associated with response, right? So, but unfortunately in India, still it is going to take time where every new outcome, genomic testing will be done. That is still.
So that's not happening right now.
It's not happening at a large level, but it is starting to happen now. So in fact, with the help of ctDNA now, what is happening is we are able to monitor the patients for their treatment, and we can catch these resistance early.
So along with your TMB score, do you also help clinicians in taking a call on which type of immunotherapy they should opt for? Because not necessarily one type of immunotherapy will work for that patient. I mean, there are so many different immune checkpoint inhibitors.
So what we do is we essentially share the data that we have in terms of other patients, what kind of immunotherapy works. So in this patient, we said, okay, this is the particular drug which was used. So atezolizumab or nivolumab or pembrolizumab, right? But this was based on not direct FDA approved. So TMB as a market is also approved by FDA. and there are specific drugs which have been approved, right? But this was not more like directly from the approval, because this way of doing the things a little differently. So it was based on the actual real world data, and that's where we're also working on building something very interesting. We are working on building a platform where a new patient comes, and based on the report, so we would upload the report. It's a earlier POC. I don't know whether I should talk about this also or not, but no, it's okay. It's not about the idea, it's about execution at the end of the day. So, but it's very exciting, that is why I want to share about it. So, it's early POC where we are taking the patient's report, we are taking the raw data, and we are looking at around 15 to 20 different clinical parameters. And a new patient comes, we first make a clinical genomic digital twin of the patient, right? And then what we are doing is, based on our database, we do twin match. And we find which other patients, at top 10 patients, have similar clinical genomic attributes. So it's not a simple Excel search. We are not doing filtering. It's not that all EGFR lung patient will show. No, not like that. It is based on a lot of data, where machine is running on its own, all these things. It is early POC, and this would be, in true sense, real world based, database, evidence based treatment. That's what we are working towards. But we are still far away from it. I think it will take some time.
This tool will love to give similar patients
Yes.
for a similar treatment line.
Treatment line could be different. So treatment line could be completely different. So the first phase, we are just looking on clubbing the patients together, see what treatment was given and which worked the best. Later point in time, it would be more in terms of, okay, now we can define the treatment line also. But right now, the first phase, it should just be firstly matching. Yeah, based on the clinical genomic attributes.
I had a question. I mean, now obviously it's science is exciting, and you now have some outcome also, as you showed. How far are you from taking it to the clinical regulatory pathway?
So this is something that we want to start now. So we are talking to some of the pharma companies, saying that with this TMB as a biomarker, with the way we are looking at the data and insights with percentile distribution than a standard single score. There's some talk going on, but how far clinical trials, you know how much time it will take, right? It will take a couple of years.
Yes.
But if we get exciting results over here, that would be fantastic, because immunotherapy as a treatment is very, very expensive. It is around 2.5-3 lakh per cycle. Patient would take 15 to 20 cycles, right? And then if there's no response, imagine, right? So this would also help in terms of identifying the patients where it might not work. There's no need to spend so much money, right? Because many patients and their families, they drain off their whole earnings and everything with the hope that patient can be saved, right? But if we can give more information which can help them take that decision, I think that would be helpful, not just from the positive outcome side, but even from the negative outcome side.
Yes. So I think that we could safely say that 4baseCare is allowing people to make better clinical decision making for cancer treatment, right?
We are trying. So our motto is simple. We are like giving cancer patient the best chance of fighting cancer. We are not saying we are going to solve cancer tomorrow. We think the approach that we have, what we are doing is, we are just trying to give the best chance. And the more we do, the better the chance becomes.
Any more questions? Faculty at the Siemens Center for the Study of Living Machines at NCBS. And his lab combines approaches of theoretical physics and machine learning and experiments to probe fundamental principles in biological systems such as circadian clock, cancer, and he has previously been a postdoc at the Harvard University and the Dana-Farber Cancer Institute, where he has worked on designing targeted therapy regimens for cancer therapy through mathematics of cancer evolution. And he has been a PhD at the University of Maryland, where he did physics on molecular motors. Maybe I can connect with you on that later, because we have some overlap there. And so today he is, after you got a genomics-based picture of how cancer can be detected, I think Shaon is going to give you a complementary picture where he will say how non-genetic origins are there in cancer and drug resistance. Over to you.
This invitation to this really nice setup. I think this is really cool, and more of this should happen actually. I love chatting with Hitesh. It's really nice. So yeah, you got a very nice overview of the kind of things that sort of modern science is able to do with genomics in cancer, and I would argue that that's the more well-researched and better understood aspect of cancer, since the Human Genome Project allowed us to figure out what genes exist and so on. But something that's not as well appreciated, but is probably equally important, is the non-genetic aspects of cancer and drug resistance. What do I mean by non-genetic drug resistance? So you can do a very simple experiment. All of you in the lab, you can do this right now when you go back. You take a dish, put some cancer cells in that dish, right? Say 5 million cancer cells in a dish, put in a drug of your choice, right? You can put in cisplatin, you can put in any tyrosine kinase inhibitor, erlotinib, imatinib, whatever you want. Throw it into that dish. What you will invariably see is that not all cells will die. Okay? You will invariably see that a fraction of cells will always survive. And this is actually something that is not new. This phenomenon has been observed since the 1940s, originally in bacteria, from where you get all this antimicrobial resistance, AMR and all of that, and you see exactly the same thing in cancer as well, okay? And you can very carefully do these experiments where you start with a single cell, let the single cell divide for a few generations so that you ensure that there are no differences in the DNA between these different cells, and you put the drug, you see the same thing, right? Some cells will die, some cells will survive, right? So it's not a difference in the DNA profile, not a difference in the mutations in the DNA that is causing some cells to survive and some cells to die, right? So there is some difference intrinsically in these cells that allow some cells to survive drug treatment, okay? And this is what we call non-genetic heterogeneity, okay? Or non-genetic origins of drug tolerance. And in fact, there's a recent paper out in Nature, I think, where they've shown using very beautiful matched data sets from colorectal cancer patients, where they show that, for example, in the primary tumor in colon cancer versus metastasis, for example, in liver, the mutational profile is almost identical, okay? So what is different then? And they argue that it's some non-genetic phenomenon, some gene expression levels, perhaps protein levels, perhaps some epigenetic aspect that is different that causes these differences, okay? So largely, these are the kinds of questions that we are interested in the lab, trying to understand why do you have differences in cells that allow them to survive versus die? Can we identify from, for example, a single sample that comes to us, can we identify what are the genes that are driving this kind of behavior? So this is one large direction that my lab is interested in. And for the first 5 minutes, I'll tell you a little bit about the kind of things we are doing there, how we are identifying certain genes and so on. And the other broader aspect of the research in our lab, as Devoshini told you, is to try and understand dependencies of cancers on other non-genetic elements that have control over cancer cells. For example, the one that we are most interested in is the circadian clock, okay? So the time of your body has a very strong influence on how cancer cells progress, on how cancer cells cycle, on the immune system of your body, and I'll tell you a little bit about how we are studying these non-genetic aspects in the context of cell proliferation, cancer, and so on, okay? So something that we have realized over the last maybe few years is something quite interesting, and this goes back to sort of fundamental biology in the dish. So one thing that we do in the lab is we do microscopy. So we image cells as they are cancer cells as they are dividing. So we can actually see how individual single cells behave before and after you put a drug therapy on it. Okay? So I'll tell you something that's really fascinating that we've been consistently observing, and I'll tell you what that means in the context of non-genetic heterogeneity. So what we see is, if you see a cell, and this cell goes and divides and gives rise to two daughter cells, okay? So this cell has divided, given rise to these two daughter cells, and if my drug treatment happened at this time, okay, what we invariably observe is that the final outcome is almost 80% of the time it's the same. So both these cells either die or both these cells survive. And this is remarkable because if you actually look into the dish, these cells might be in completely different parts of the dish, okay? Yet, somehow they have a knowledge of what its sibling is actually doing. So if this sibling dies, that sibling also is very likely to die. If this sibling survives, this is also very likely to survive, okay? That's one observation. The second observation is, if instead of the drug being administered here before these cells were born, if this was administered at this point, which is after the cells are born, okay? So you have two independent cells which have never seen drug, which are moving around in your dish far away from each other, and now you've added the drug, you still have the same fate 80% of the time. Okay? This is rather remarkable. So somehow, magically, these cells, sibling cells, know what the other is doing, irrespective of when you've added the drug, here or here. Okay? What this tells us, the broad picture, is that these cells are inheriting something from its mother, which allows it to either survive the treatment or die in the face of the treatment. Okay? So that word is now becoming increasingly important in the field of non-genetic drug tolerance, which is inheritability. Okay? Inheritability or inheritance. So there's now a lot of research coming out which is suggesting that something, we don't know yet what that something is. It's not anything to do with the DNA, as I've argued before. Something in these cells, which we broadly call cell states, that is inherited from mother cell to daughter cells to granddaughter cells, and so on, which prime these cells to take a particular cell fate, death or survival. And these states exist well before you've even added the drug. Okay? So there is a notion that you add a drug and the drug induces some kind of fate in your cells. What we are suggesting and what the field is now beginning to understand is that may not be completely correct. In fact, the cells are pre-programmed to already sort of be primed towards a particular fate or towards survival or death. Okay? So that's one big understanding that has come into the field in the last maybe 10 years. Okay? That inheritable cell states are what is driving a lot of the non-genetic drug tolerance in cancer. So the second question then, the obvious question is, what are these states, right? I mean, it's one thing to say that you have inheritable cell states. What are these states? So this is a collaboration that I have with a close colleague of mine, Archishman, in the Simon Center, where we are asking, can we use this information of inheritability to find which genes expression levels are associated with this kind of a phenomenon? Okay, so I'll give you a very brief idea of how we are doing this, and this is very powerful because for the first time we are able to take a single sample. So say a patient walks into the clinic, gives a single sample of blood or a tumor biopsy. From that single sample, if we can do single cell RNA-seq, which has now become very easy to do, we can actually pull out information of what are the genes, okay? So for the moment, we have not looked into anything else. It's true that it could be, say, protein levels, it could be histone modifications, it could be DNA methylation, but there is some evidence to show that gene expression can be driving this kind of a phenomenon, and we are able to pull out which genes are driving this phenomenon, okay? How do we do this? So I'll give you a very brief outline of this because this also involves a lot of computational aspects, physics aspects. So if you get a sample, say a tissue sample, and you do your standard single cell RNA-seq, the data that you get looks like this, okay? So this data will have cells along this direction and genes along this direction, okay? So each entry corresponds to the gene expression level of a particular cell, okay? So it turns out that if in this data, so remember in this data, the question that we are asking is, which of these genes are giving rise to this inheritance phenomenon, okay? Now you can sort of already appreciate that this is a tough problem because inheritance is strongly related to a temporal phenomenon, right? Over time, over cell divisions, something is being inherited, right? So it's a time-related phenomenon. Here, there is no time involved because you've taken it from a single sample, right? And yet you are trying to pull out some information of time, right? But how do we do it? So it turns out it can be done. The reason it can be done is what we have shown in this work together with Archishman and a student who's sitting right here, Shuvranil, is that if you have these kinds of inheritable genes in your sample, okay? And these are being called memory genes, okay? So if you look this up, you'll see that this is a new subject. It was originally coined by Arjun Raj's lab in University of Pennsylvania. So if you have memory genes in your data set, it turns out that there is a very interesting structure in what is called the covariance matrix, okay? So from this matrix, you can construct what is called a covariance matrix. For those of you who are more inclined towards bioinformatics or computer science, you would have heard of something called principal component analysis. In principal component analysis, you construct this covariance matrix and you look at its eigenvalues, right? So what it turns out is that if you have a data with memory genes, then the covariance matrix will have a very interesting structure in the eigenvalues, okay? So if you plot the rank of the eigenvalues versus the eigenvalues themselves, you will see this structure, okay? So leaving aside the technical aspects, all I want to say is that when you have these memory genes in your data, you see a particular kind of structure in the eigenvalues. So this is something that we've been using to actually identify which genes are these so-called memory genes. Because what we do is we take out a single gene at a time. If that gene was a memory gene and you've taken it out, then this structure gets lost, and what your data will then look like is something like this, okay? So using this technique, we've come up with the first method that allows you to take a tissue sample, from that single sample, measure the gene expression, and figure out which genes are these so-called memory genes, okay? And the hope is that once we are able to do this better, we will be able to identify from just single snapshots which genes are driving this kind of tolerance to drug resistance, okay? So that's broadly one aspect of what we do. The other aspect which I told you is to look for dependencies of cancer with other non-genetic aspects of cancer cells. And this is where we are working a lot with the circadian clock. And for those of you who don't know, the circadian clock is essentially what allows us to synchronize with the day and night cycle outside. Okay? Now it turns out that all the single cells in our body, including these cancer cells, if you look at their single cell gene expression profile, typically have a clock that oscillates like this, okay, with a 24-hour time period. So these are gene expression profiles or proteins of the circadian clock network, and if you follow them over time, so this could be some kind of gene expression level, okay? It oscillates like this. Now, what we have shown, and others have shown as well, is that if this circadian clock is connected to the cell cycle of the cancer, remember, cancer, once again, the cells are proliferating rapidly, these are fast dividing cells, and so the cell cycle is an important component of any kind of cancer, right? So, so if the circadian clock is connected to the cell cycle, and this people have shown before, links have been found, molecular links have been found between the circadian clock and the cell cycle. What this would imply is that if you look at the fraction of cells in any particular cell cycle phase. So I'm sure all of you know that the cell cycle, you can have G1, S, G2, M, and so on. If you look at the fraction of cells in any phase, say in M phase, normally you would expect a flat line, okay? That a lot of cells are in the G1 phase, fewer cells in the S phase, and so on. But if you have this coupling to the circadian clock, what you can show theoretically is that this fraction will now oscillate, which means there are certain times when the cancer cells are proliferating the most and certain times when they are proliferating the least. Okay? So that's one point. That's an important point. The second is that, so this, let's say, is the cancer cell. The cancer cell, fraction of cells in a particular phase is doing this, okay? Now, the same circadian clock is also present in the normal cells. So for example, in the intestine, people have shown for a very long time that you in fact do see these kinds of 24-hour oscillations in proliferation patterns in all through your intestine, okay? What we have shown is that if the same circadian clock is coupling to the normal cells, which has a difference in the cycling time, right? So let's say this cancer cell is cycling with 16 hours, which is, for example, in the lab, this is typical of colon cancer cells. But say the normal cells are cycling with 24 hours or 30 hours, right? These are slower because these are normal cells. What we can show is that the oscillating pattern of the fraction of cells in a particular phase will actually look phase shifted. Okay? So that will look like maybe this. So this dashed line is for a normal cell. What this means is we have what is called a therapeutic window that has developed here. This is a therapeutic window, which means that at two different times will you see proliferation peaks of the normal cells versus the cancer cells. So what this means, and this is a huge field now that is coming up, which is called chronotherapy, is if you can identify these correct times, then what you would want to do is you would want to administer your chemotherapy at this time when the cancer cells are at its peak, but the normal cells are at its trough. Okay? So this way you would hope to be able to deliver the drug with the least amount of side effects. Okay? So the big question in the field obviously now, and this goes back to the kind of things Hitesh was saying about personalizing cancer therapy, is to be able to figure out this time of the patient. Okay? It turns out, and this is again sort of understanding that has happened in the last maybe 20 years, is that each of us here have very different body times, and you will realize this immediately because some of you love waking up early in the morning, but if you're like me, you might hate waking up early in the morning. This is directly connected to where your body clock is speaking and when it is troughing. Okay? So depending on that, your body time can be quite significantly different. It can be as different as 12 hours. Okay? So we know theoretically that we can generate these therapeutic windows because the circadian clock couples to cell cycle. But the big challenge in the field right now is to be able to figure out at a personalized level where you are in this circadian oscillation. Okay? And that's another big direction that my lab is working on. And for example, Anjum here is working a lot on this. The whole idea being, you get a single sample from a patient. We haven't reached patient levels yet. We are working on tissues now, but you receive some sample, maybe from a tumor, maybe from saliva. From that, we want to be able to figure out where that patient is on this circadian oscillation. Okay? And with that, the hope is one can do some kind of chronotherapy. Okay? So maybe I'll stop here for questions.
Questions?
Thanks for the nice talk. I have a question in the first part of your talk where you talk about memory genes. The first question is does that hold good for all cancer cells? Like, for example, why I'm asking that question is, does it hold good even for glioma? Because there's something which peaks up very quickly after certain stages. So it would be nice to know the memory genes identified so that you can have a good cure about it. Second question is, can this kind of memory genes identification, can you kind of extrapolate for general health? You know, that you can predict, oh, this is the disease which I'm going to get. It's either a survival or a death mode for me. Would that be I mean, this is a little crazy question. Can that be extrapolated? If memory genes are known for each organ or some failures, because these days sequencing has become quite cheaper for every common man. So can we kind of extrapolate to understand, am I going to die? So things like that. Thanks.
Okay, so let me answer your first question. That's, I think, easier to answer. So almost definitely, even if you take glioma cells in a dish, you add drug, you'll see this fractional killing effect for sure. Right? That some cells will die, some cells will survive. Now, whether there are memory genes there, I am not aware that people have really tried to look into that because it's a really new area, maybe just 4 years, 5 years old. So the place where this has been mostly studied is in melanoma. So we've also, the kind of data sets we've studied has been on melanoma. And in melanoma, at least, we see that the kind of memory genes that come up include things like AXL. I don't know if any of you have heard of this particular gene. It's a very famous gene that's related to extracellular matrix and then plasticity of cells. It's also a targetable gene, and so it's very interesting that AXL comes up as a memory gene. Other genes like EGFR, NGFR, fibronectin, these also come up as memory genes. So we know much more about these memory genes in melanoma as compared to anything else for the moment. But yeah, it would be, because now we have this method, I think it would be very easy to go and just explore in other cancer types. About your second question, I have no idea, right? I mean, I don't think anyone has ever associated memory genes with anything else. As of now, to the best of my knowledge, I think this has been associated only with non-genetic drug tolerance in cancer and potentially in AMR, right? So because a lot of these concepts are fundamentally related with bacterial drug tolerance, in fact, pretty much all these ideas originally came from bacteria and antibiotic resistance. So my hunch would be a lot of this would hold in bacterial and antibiotic resistance. Yeah, but beyond that, I don't know.
There are biomarkers available which predicts the disease. So,
maybe, yeah, maybe it's possible. Yeah, but we haven't looked at this yet.
So all these memory genes which you just mentioned, will it be common to different types of therapy resistance? Say for example, radio resistance, chemo resistance, or say for immunotherapy resistance. So will these sets of genes will be common, and how do you plan to extrapolate this information for cracking the resistance problems in patients?
This is an excellent question. So the immediate answer is we don't really know, but what we have some evidence for is that a lot of these memory genes that we are getting is associated somehow with lineage relationships, right? That these cells are close to each other in lineage. And what is also happening is that we are seeing some evidence of extracellular matrix and microenvironment kind of gene expression that we are seeing coming up with these memory genes, right? So as a result, my hunch is that a lot of these genes will be common between different cancer because it has ultimately got to do with the extracellular matrix, things like fibronectin, overexpression. And so in a completely separate analysis, we do see that anything to do with lineages of which cells are close to each other has common genes coming up, right? So my feeling is, yes, that a lot of these memory genes might actually be common between different cancers. But as in my response to her question, it's too early to say. It's really just fresh off the sort of oven, and we've not really tested these things yet. And in answer to your second question, though, people have already started doing this. I think Arjun Raj's lab has already been playing with this idea a bit. If you see some memory genes whose expression levels are going high, which is leading to the drug tolerance, then you can simply do some kind of knockdown, or maybe not knockout, but knockdowns, maybe some siRNA kind of thing where you reduce this gene expression levels temporarily, right? Which allow you to then go and hit the cells with the same drug, and you see a lot more cell death happening. So this in fact has already been demonstrated, that you just take this memory gene, you reduce the gene expression, you treat it once again with the drug, and it turns out that you get much better cancer cell kill, right? So that is the way I think forward.
And on what basis you classify those genes as memory genes? Because when you try to compare resistance versus control population, you will see multiple, n number of genes getting overexpressed or downregulated.
Right.
On what basis you are classifying?
Yes. So at the heart of the definition of a memory gene is this notion that it has to be inheritable across many cell divisions. So this is not true about most genes. So if you look at the gene expression of a particular gene at the single cell level, it actually fluctuates a lot, right? So from one cell to another cell, it could be very different. So if you track the expression levels of a particular gene across many cell divisions, if that gene expression level stays high in the mother, in the daughter, in the granddaughter, and so on, that is a memory gene. And that will not happen for most of the other genes. Okay? So that's why this is called a memory gene.
Does it have to be in response to a stimulus?
No, this is all happening before you've added a drug. So this is once again going back to this thing that I was saying before, that these states, these sort of high expression states, all exist in the cells well before you've even added the drug. So therefore, in some sense, these cells are already primed to behave in a certain way well before you've added the drug. Okay, so once you add the drug, it just responds according to what state it was already in some primed state. So this is again a very interesting notion, that it's not the drug that is really driving things, it's the state of the cell that it already existed in for the past maybe 4 or 5 generations.
My question was not really the stimulus, the drug being the stimulus, but rather some other
Absolutely, absolutely. So that would absolutely be the case, that your microenvironmental niche might be providing some signals that put a particular happening well before the drug. So the microenvironment absolutely, I think, will matter.
I'm still a little confused about the concept,
Yeah.
but some of the genes could be consistently down, which could lead to inheritance. Some could be consistently up. So how can we say just the upregulated ones?
Again, very good question, and this is something we have been grappling with. It's an excellent question, because when gene expression is down, it's very hard to say, at least with our current technologies. I don't know how many of you are doing single cell RNA-seq, and I hope I don't want to offend anyone, but single cell RNA-seq technology is terrible. Okay, it's terrible because if you have 100 mRNA in a cell, the best current technology will capture only 30 of those RNA. You will lose 70 RNA. That's the best technology. Even most technologies will get you 10% of the RNA, right? So in your data, if you see zeros or very low expression of a particular gene, there is simply no way of saying whether this is a technical error or is it a real thing, right? So as of now, we have sort of really focused only on the high expression states, because with single cell RNA-seq, you just cannot do that. It's just, yeah.
So I have a background in systems biology, and I was into network biology for a while. So there, if something is getting suppressed, there would be a mechanism to impose that suppression. So that mechanism, so that's where network biology might also play a good role.
Absolutely.
In showing that, okay, some mechanism is up, that may not be itself an inheritance, but the actual inheritance property is coming from what is being suppressed.
Oh, absolutely, you're absolutely right. So it's almost sure that you have low expression states that are also being inherited.
Yeah.
And there are also high expression states that are being inherited.
Yeah.
For the moment, we are only able to detect the ones which are high, right? Yeah, exactly. Yeah.
And these are falling into different classes, like you mentioned, AXL, tyrosine kinase,
Yes.
kinases. Then you've got receptors, and
Yes, exactly,
all of those. There's different
exactly.
classes. So you're looking at the expression levels and then trying to classify and then identify what could be a possible memory genes, and then you're trying to kind of suppress it and then identify if that is leading to a change in the fate on an external stimulation.
Correct, absolutely.
Okay.
That's a perfect summary. Yeah.
Okay.
A good number of mitochondrial genes also.
I have to check. I can't remember.
It is by consent from mother to
So but I can't remember having seen mitochondrial genes showing up.
That's something that's common across all, mother to daughter.
Yeah, but this you have to remember is at the level of the gene expression. This is not the genes themselves, right? This is expression levels that have to be inherited, right? So if it's high in the mother, it has to stay high in the daughters and the granddaughters and so on. So we've actually not seen any mitochondrial genes yet.
So this kind of behavior in single cell clonally expanded populations is easy to understand, but in a real world scenario you are likely to encounter heterogeneous populations within the cancer cells. So this therapeutic window concept, how tight the correlation in terms of their this oscillation profile would be. Any insights till now?
Not directly. It's a good question, because definitely there's going to be a lot of heterogeneity in the cancer. So I don't know yet. These are the kinds of questions that we are moving towards. But we do know that in melanoma, for example, at least in mice, there have been people who have shown that if you track the proliferation of cells as a function of time, right? So because in mice tend to be a little bit more homogeneous, right? You have these 12-hour light dark cycles that are given to mice. Once you inject mice with melanoma cells, people have shown that the cell proliferation in skin cells of mice do actually show this kind of very nice behavior. So at least with mouse models, we know that there is this kind of an effect. How do we do this? How do we show this in patients? This is a much harder problem. And to do that, the first thing that one has to be able to do is to actually time the patient, right? So figure out what is the position of that individual patient on these oscillations, right? And then one would have to look at the cell cycle status of the cells in the tissue of that patient, right? So these are definitely open questions. There are no answers yet, but that's where we are trying to move. Yeah.
Just from this heterogeneity perspective, right? So one of the ways we try to manage tumor heterogeneity, because there's a problem everywhere, is we try to work with, say, even FFP tissue and even ctDNA. Do you think the profile would be similar if you use tumor tissue or, say, enrich ctDNA and do the same?
So once again, this is a good question, because what we are finding out now is that there are contradictory reports coming in about what these profiles look like in tumors, right? In fact, what some people are suggesting is that in tumors, this profile may actually become completely flat. And so one thought is that if these profiles become completely flat, what one would have to do is to actually bring back these profiles into an oscillatory mode, right? So what you are saying is absolutely right. It's a very, very complicated scenario. What these profiles would actually even look like is something that we don't fully understand. In certain cancers, for example in colorectal cancer, people have shown that these profiles are existent, right? So you see these very nice oscillations. But in some cancers, these profiles are completely lost. So what do you do about that? So then you have to try and use these small molecule activators of the clock. So there's a huge amount of research going into building small molecule activators. And the idea and the hope is, if you see that these profiles have gone flat, then you first add some small molecule activator, get them to oscillate like this, and then do your the chronotherapy. But again, it's very complicated, so no simple answers.
First part, I just wanted to understand the first one that you said, that you will start with one single cell, and then it becomes two, and then if they are coming from the same mother, their behavior is going to be similar, right? And from another cell, they may have either you call that as tolerance, if suppose, or susceptible. That is the first category that's the base assumption, right? So if you are using a cell and you say that to be called as a memory gene, you have to see the expression for at least 4 generations. At least.
Yeah, 4,
right?
5 roughly, yeah.
So starting with the same cell clone, which usually whenever we are doing cell culture, most of them have been isolated as a single cell clone, which have been propagated in multiple labs over multiple generations. So here, after say 4 generations, the same cell may turn to be susceptible, right? That will be depending on the memory gene's expression up and down.
Absolutely.
Assuming that memory gene is the primary thing which is driving, which would be other mutations acquired transcription things. But for just understanding for this particular assumption, up to 4 generations, if the levels are remaining high, it is susceptible or tolerant. But the same cell may change the nature with a couple of other divisions.
Yes, absolutely. And it doesn't have to be exactly 4. What we are seeing is it's maybe 4 to 6, 7. Yes, but you are absolutely right. The same particular cell, when it's in a particular high state of these memory genes, is likely to be tolerant to a drug if you add the drug at that time. But if you wait for longer and the cell divides a few times and it switches from that high state to that low state of the gene expression of these memory genes, and then you add the drug, these cells would die. So absolutely right. So
why I said this is
Yeah.
because in practical experience, where we have seen it in different cells, sometimes there is human which are blood cancer cells and lung cancer cells. Where we were looking at some receptors on the surface. So even though they have started from the same cell, even when somebody has reported that at least 30% of these cells should have these receptors. But we were looking into our groups, which have come from the same group, because of course passage multiple times, where by genotype may remain same, their phenotype has changed completely.
absolutely. And the interesting thing is, that phenotype, if you keep watching for some more time, you might see it coming back. So this is the sort of sign of these memory gene expression states.
Also, when we keep passaging the cells, sometimes if you're looking at, say, drug susceptibility, if you do it at, say, third passage, when they are at a certain stage, you'll see highest effect. You let it passage for, say, 10 times, you will not see the same impact.
Absolutely.
So that happens with the same culture, same clone.
Exactly. And this is, I think, we are beginning to understand why this might be the case, and to also be able to identify the genes that are potentially responsible for this behavior. Yeah, absolutely.
So in addition to the heterogeneity of the cells in cancer, the microbiome also plays a very crucial role. Now there are reports which are coming that microbiome has an impact on cancer progression. So do you think the predictivity of your model or the circadian rhythm that you're predicting has any correlation with that for the microbiome?
So I have no expertise in the microbiome, but I have talked to a few people, and I know that the microbiome itself is under very strong effect of the circadian clock. So depending on when you measure your microbiome, you will actually see very different levels of bacteria depending on the time when you actually sequence it. So yeah, but I mean, again, beyond this, I don't know anything about the microbiome, but it will definitely have an effect, right? But once again, so it boils down to us being able to actually measure what your body's clock time is. That is becoming more and more important, right? Because that determines a lot of these things, whether your microbiome is high, low, whether your immune system is high, low, whether your DNA damage response is high or low. So that's one of the big challenges, I think, in the field today, measuring your body time accurately. And in fact, just to add to that, a lot of what is happening, or a lot of what people are trying to do, we've not got into this yet, is using wearable devices, because now literally everyone has some Fitbit something, right, which is constantly tracking your heart rate something, right? And so another very exciting field, I don't know of anyone doing this in India, but in other universities, where you take all of that data from your Fitbit and you try to actually figure out where you are on this circadian clock, right? And based on that, you will then do all kinds of other things. We don't know. There have been contradictory reports. So there's no clear answer yet. In fact, in some reports, people have said that having this kind of an intact oscillatory structure is very important for the cancer itself, right? So the cancer is surviving on this kind of an oscillation, whereas some reports have said that it's bad for the cancer. So it's very early days, so we really don't know what is going on. And this is all things that one has to explore over the next few years.
I think now we will call it an end to this edition of Shubha Rush with Science. Thank you, Shaon, and thank you, Hitesh. Amazing. I think you all agree it was an amazing session today. So with that, I think we will open up the ice cream. Yes, please help yourselves. And thanks again, Shaon and Hitesh.
Very nice.